All right, welcome everybody to the fourth week of the virtual Monday launch. Bill Hill, our president's got a few words to say before we get started.
Howdy, Aggies. My name is Bill Hill, class of '84, and your San Antonio A&M club president. You know, we haven't been meeting at Aggie Park lately, and we've been adapting and innovating. And so that's kind of what I'm doing here today. I'm out actually on the golf course. I just rounded the turn at 9. I'm on 10 at Fort Sam Houston. In the background, we've got the Fort Sam Houston Cemetery. I don't know if you can see that, but just as a reminder, this is Memorial Day weekend, and on Memorial Day we pay tribute to those soldiers who lost their lives in combat. And based upon last week's program, where we had Bronson Burgone, class of 2009, PGA Tour member, and a member of the 2009 Fighting Texas Aggie National Championship golf team, it motivated me to get out here on the golf course. So we got a great program ahead. Logan will lead the rest of the way. I've got 2 announcements. Number 1, Aggie Family Barbecue has been rescheduled, and it's currently scheduled for July 18th. There'll be more information to follow on that. The San Antonio A&M Golf Tournament is scheduled for September 10th. Again, information in the Alamo Aggie, which will be coming out in about 3 weeks, should have the details associated with that. So anyway, that about wraps it up for me here, Logan. I do want to say that we did continue to get contributions for our capital campaign. And currently right now, we stand at $935,100. That's an increase from last week. And again, our goal is 1.6. Logan, I'm going to turn it over to you, and I'm going to get back onto the game. Giggle.
All right, thanks, Bill. Play well.
All right.
All right. Excited this week to get to speak to Dr. Ben Neuman, who is the head of the biology department of Texas A&M Texarkana and a coronavirus expert. So well respected. You were on the international committee that named the SARS-CoV-2 virus. So welcome, Dr. Ben. How are you doing today?
Hey, great to be here. I'm doing all right. Yeah. How are you?
Doing good. Thanks for joining us. I guess to get started, when did you begin studying coronaviruses, and how'd you get into that?
Yeah, it was kind of an accident at first. I did a year abroad, and when I came back, you had to do a senior project, and they had all filled up except for 2. One was working on a virus, the other was working on a beauty cream, the science behind it, which it turns out there wasn't a whole lot of science behind it. It just feels nice on your skin.
Yeah.
That virus job turned into a volunteer spot at the med school, and we were working on the mouse coronavirus, and that was my first time growing a coronavirus back in '96. Yeah, it's been a while. Yeah. More than half my life, I guess, has been working on coronaviruses so far.
You've been getting ready for this moment for a long time, it sounds like.
Yeah, that's right. I've kind of always been stuck on coronaviruses. It's neat to see everybody talking about them, but I wish it was not in this context. Yeah.
Definitely. About this new coronavirus, there's just so much information out there, and as more and more information comes in each day, what are some of the perceptions that have changed since we first started talking about this a few months back?
I mean, perceptions have changed, but also just our understanding of this virus has changed. So before, you would go to a coronavirus conference, and there are maybe, 150 generously people that actually work on these things full time. And you see the same faces every 3 years, that's fine. Now it seems like all of virology and quite a bit of all of biology has shifted over to focus on this, and the progress is absolutely amazing. There are things I've been waiting on my entire scientific career, and they just keep coming. You'll get multiple structures in a week where we did not get more than a structure every 2 or 3 years before. And it's completely changing how we understand the virus. We can just get much closer to this one. We see how it works a little bit better. Coronavirus is finally joining things like HIV that we actually understand relatively well. Now, actually finding a way to stop it, that's probably the next step, but we're not quite there yet.
Well, HIV took a couple decades to get a vaccine for. How's it looking?
Still don't have a vaccine.
Still don't have a complete vaccine.
Lots of
Is
trials, lots of good ideas, but they have all failed. Yeah.
is that something that we could be looking at with this with the SARS-CoV-2?
That's the thing we don't know, but at this point we haven't really tried. And so thank goodness, yeah, we got room for hope still. And there are a lot of good ideas out there. There are over 100 vaccine studies registered with the WHO in various stages. I think there are only 5 of those actually in clinical trials, although maybe a 6th one has joined now. But all those other ones are on their way. And if the first couple don't work, there are plenty more good ideas out there. And I feel like at least one of these is going to do something. Yeah.
Well, let's hope so. We've got a question from Ken King, class of '60, and he was wondering if if you ever worked in Wuhan and how was
Never
it?
worked in Wuhan, but worked with a guy called Yu Chen, who's a professor out in Wuhan. We collaborated on a shrimp corona-like virus. He was trying to look at some of the enzymes of this and figure out exactly what they did and prove it. And I was good at bioinformatics. And so I looked at the thing and said, no, you're actually trying to make a protein and a half there. You want to cut it off here to get just your protein. And then it's more likely to work. And it turns out it did work, and that was great. And then we collaborated on a sea slug corona-like virus. There's so many things related to coronaviruses out there, and they're really all around us. They spill over into humans every once in a while, and we notice them. But oh my goodness, yeah, they're pretty big in nature.
So the other two that spilled over, SARS and MERS, are those the two?
Actually, every single one of the human coronaviruses has an equivalent, and I believe they're all in bats. So there are bats in this country that have things related to the human common cold viruses, which is to say they have viruses that there's a reasonable chance they could grow inside of a person. And these things grow really well inside of people. The difference between human mild coronaviruses and severe ones is less than you would think. They actually both grow to about the same level. They both enter your cells. It's just that one provokes a slightly different immune response than the other. And that's the thing that we don't really understand how it's happening. And so we need to get good at dealing with coronaviruses because there are going to be more, almost certainly.
Where has SARS and MERS gone? It was, you just don't hear much about them anymore.
Right.
So what's going on with that?
All right, so MERS first, because that one's easier. MERS comes back once or twice a year. There's an outbreak every time the baby camels are born. We've now figured out, though, that it's coming from bats into camels, and the adult camels are passing it to the baby camels, or the baby camels are eating bat poo or something like that and getting infected. And then from there, it's the people that have contact with the baby camels in particular that are the ones that are getting infected. It's over 50% of the cases now that we understand this are coming from that. And so we've been able to limit out the outbreak because we know where the source of the virus is. And so, yeah, I guess somebody has to tend to the baby camels, but at the same time, you can take precautions that will very much limit the spread. Now, original SARS came out. It was very big in 2003, came back again just a little bit in 2004, and we managed to contain it, quarantine it, and it died out in humans. But people have been looking at bat viruses, and every time they sequence a virus from another bat over there, it adds to this pile. So we have at least 50 of these SARS-like coronaviruses that we know now. And there was a study that looked at them, and it turns out that about a quarter of those are perfectly able to grow in human cells. It's just that they haven't had the opportunity yet. But nobody tells bats where to sleep at night, So yeah, this is concerning, and this suggests that there's a lot more. And there are also a lot more viruses related to MERS in bats in Africa and the Middle East. And the SARS-like coronaviruses are also found in Africa, all of Asia, and parts of Europe. We know we have bats with coronaviruses here in the US. I actually haven't seen any studies on the viruses of bats in Texas. I would be interested to see that because every time people look, they find more coronaviruses, that's for sure.
I was about to ask you, there's so many places in Texas you can go to watch the bats come out at night, caves and under highways and just, But there
Yeah.
hasn't been any studies on the viruses in those bats yet.
No, I think they're mostly worried about that white nose disease that was knocking the bats out, but not that I have seen. There was a study in the Appalachians, North Carolina lab did that. There was a study in Colorado on bats. I believe those were separate. And the rest of the country is mostly unknown. And each of those was a while ago, and technology's improved. And yeah, it's really anybody's guess until we go and look. And that's the thing. The more we look, the more corona-like viruses we find, and proper coronaviruses. Yeah.
Oh, so are we currently trying to trace back to where the SARS-CoV-2 came from? You know, like it found it with baby camels for MERS. So what's the process on that?
Yeah. So the process is a couple different threads that come together in the middle, hopefully, and all point to the same answer. So first of all, you've got the actual cases. Now, like I said, with MERS, about half of the cases had had contact, intimate contact with camels, like camel feeders, carers. These things are racing camels for the most part. And so they are very expensive, and they are people's, you know, prized little babies, just like a thoroughbred would be, just a weird looking thoroughbred. And so it's the same thing with this virus. About half of the early cases were directly associated with a particular animal market. And the other half may well be people that were sort of one person distant from somebody who was at the animal market and a case that wasn't ever discovered. So it looks like it's there. And there are now 3 clusters of these viruses. And each cluster has many, many members. When I say there are 50 types, I mean 50 species. But a species can have hundreds of little isolates that are all a bit different from each other inside of it. Yes, there's really a lot of them. The outermost ring, if you like, is all viruses of bats. And they're viruses of these things called microbats that are about this big and just adorable little things. Mostly horseshoe bats, which have a, it's not like a horseshoe, it's really got a point at the top, but that's the thing that's over their nose. And there are a lot of different kinds over in China, and this virus is very widespread. And then the next level down, getting a little closer to our virus, is all in pangolins, which are like a scaly anteater, I don't know, sort of looking thing. They're adorable. But they've isolated this from several pangolin populations. It turns out that all along, pangolins have had viruses that are almost identical to this coronavirus and have proteins that would let them grow in humans. It's just that they aren't widely kept as pets. You're actually not allowed to trade in them at all. And so probably that's what's been saving us to this point. Thank goodness, yeah. Gotta watch those pangolins. Anyway, well, the last level down is a bat virus. It's only about 1,000 nucleotides different, but this is a virus that changes at a rate of about 30 nucleotides a year. And so you're still talking something like 100 years worth of evolution, separating that virus from the one we've got now. And we haven't found anything any closer than this. So people have a lot of theories about, you know, is it a recombinant? But until you find the 2 pieces that were put together to make a recombinant out there in nature, you really can't say that with any certainty. And so, yeah, we need to go out and sample more animals in and around Wuhan, or the type of animals that are brought into the Wuhan wet market.
How big is that wet market over there?
It's bigger than your average farmer's market because it's a really big city, but yeah, it's the kind of the same principle. People are bringing in produce and a lot more live animals, I guess, than you would get. They normally don't butcher the cow right in front of you yeah, at ours, but yeah, that's the idea.
Well, we've got a question from Sherry Sowell, class of 95. She has a son starting at A&M in the fall, and she's a little nervous, and she was hoping you could speak to parents with regards on how concerned they should be with their kids going to a university or to a school or a setting like that during these times.
Yeah, I know what you mean. So what I see happening around the country is that universities are getting ready to go back. But you got to take that with the grain of salt that getting ready doesn't mean you're necessarily going to do it. And if they didn't get ready for it, then there's no way we could go back. And so at least if you prepare ahead, then there's a chance, you know. I think every university in the country has got a finger on the eject button, just in case things get a lot worse. Because we've dealt with, teaching half a term online, and honestly, I'm not a fan at all, but it can work and it can be effective. In terms of where would the high risk be? So, something like the football team is fairly compartmentalized, and as long as there's a lot of testing and they keep them kind of sequestered in the same place, there's actually a way that you could do that safely. Now, being in the stands, packed in there on game day, oh, man, yeah, that sounds like a whole lot of risk. So the study came out yesterday. What we learned from that is that 80% of the cases have been spread by 9% of people. And it's not people that are, weird in some biological way. It's people that were all adults going to large, close gatherings, typically indoors, although there have been some big ones outdoors as well. And so, yeah, the risk is especially high in something like a crowded classroom or, well, like a club or a bar. That would be extremely high. So tell your kids, stay out of the bar. Yeah.
I think most parents say stay out of Northgate, but it just doesn't go over well with the students.
Yeah.
That's a tough one to take.
I'm afraid so. Yeah.
What are your thoughts on the new CDC guidelines for reopening the schools, and does it seem realistic?
So I like that they are planning, but right now it only looks like half a plan to me. So we've got plans for exactly how to go forward, but we have no plan for if cases start to spike and we need to pause or change direction. We got a kind of a one-way street built, and we're not sure exactly where that one-way street heads. Right now in Texas, there's been a kind of a spike, sort of an uptick in cases since we ended the stay-at-home order statewide. But it's not quite as high as it could have been if everybody was being completely irresponsible. So at least there's that, right? We found out that masks are extremely effective, and boy, I know people don't like them, but man, do they work. And with a mask on, you can basically do most of the things that you would have done before, and it's safe. It seems to me like just a completely small price to pay, like personal annoyance, to be able to actually go out and experience life as it used to be in the time before Corona. Yeah, the BC. Yeah.
So are you saying we should be able to get back to normal, just wear a mask?
For the most part, yeah. That's the only thing that an average person can do. The other things all have to happen at the state or national level, because you're not going to personally build your own testing lab, and roll out more of those or set up your own contact tracing network. You may call your family every weekend, but it's got to be a little bigger, a little more coordinated for that. These efforts are starting. I think we've been a little slow on actually getting those rolled out, which is why the problem has got so huge here. But it does, at least to me, in my professional opinion, it does still look like something that we can stop, hopefully by the end of the year in the US. And the most optimistic models show it going right down to fairly low levels by the end of the summer. But right now, the cases are rising rather than falling in a lot of places. And until you see them turn around, I can't have a lot of confidence in those models.
How many people, how accurate do you think those numbers are? We've got a lot of positive tests, but I've heard so many people say that there's so many asymptomatic people.
So
it seems like that number might be a lot larger.
The way you can actually check this is that we know from all of the really controlled studies, like when you had that cruise ship and everybody on board was tested and they caught every single asymptomatic and they kept them on there for a month until everybody was over it or dead. From places like that, we know that the actual fatality rate is somewhere around 1.3 to 1.5%. Current fatality rate in the state of Texas, I think, is somewhere around 4%, and nationally it's more like 6 or 7%. That doesn't mean the virus has got more dangerous. It just tells you how many cases we're missing. So if the real rate is 1% and we're reading out 4%, then there are 3 cases out there for every one that we don't know about. And it's easy to count dead people because they don't go anywhere. It is harder to count asymptomatic people because you can't tell who they are. Yeah.
Definitely. So So what, I guess it's you're saying that the death rate is, as far as you can tell, is like 1.5%.
Right. And that's what it should be. And if you see a higher rate, you just, you fill in the missing gap with cases that we don't know. And there are different statistical ways to do it, but that's like the simplest one that anybody can just look at the number and say, oh, okay, I know about how many we're missing. And so far, those turn out to be fairly accurate. Those predict kind of the shape of long-term rises and falls in different countries. So yeah.
If we just let this virus run wild, it's already in like every corner of the world.
Yeah.
Is it like, would it infect everybody if we just didn't do anything? Or is there like a...
Well, all right, yeah, two things. One, from what we know about human coronaviruses, your immunity usually lasts a little longer than a year, but less than 2 years. So you're looking at maybe getting this thing again with a 1% chance of dying every second or third year. And compare this to other diseases that we've known about, like something like polio. I think that only paralyzed about 1 in 1,000 people and killed far fewer than that, maybe 1 in 10,000 people. Here we've got a virus that kills 1 out of 100 people. So it's essentially 10 times worse than polio. And we have never seen in human history a modern society have to deal with an outbreak of this proportion, this magnitude, and this death rate. This really changes everything. And it's, I mean, it would be inconceivable to go back and say, yeah, sure, just let polio burn. I think the difference there was that you had all those crippled people afterward and you could see them. My babysitter as a child had caught polio right at the end, just before they eradicated it in Arkansas. And yeah, it was visible evidence. Whereas with coronavirus, people just die, yeah, and then they disappear. And so you think, oh, everything's fine. But really, it's it's not.
Well, I was not trying to disrespect anybody. I mean, that has passed away or has gotten the disease.
No, yeah.
I think
this is not you. This is everybody. Yeah.
I was just curious of what the infection rate is. I mean, would it infect 100% of the population, or does it, or does only 40%, because it's a 1.5% death rate, but of the people that actually get it. So
it depends. It depends on what people do. There was a really neat study that came out where they traced. So they found a guy who was working at a grocery store, and he was interacting with lots of people going through the checkout line, and also with colleagues. And the rate of just regular people coming through the store that caught the virus was extremely low. It was something like 0.05%, just almost negligible. And some of those could have caught it somewhere else too, because there's, plenty of virus around. For coworkers, it was actually 9% chance that they would catch it. And so that's working with a person. And we also know that the virus clusters tends to be within a household, you'll get the spread. And you got about a 12% chance if somebody in your house has the COVID-19 that you will also catch it, which is not as high as you might think. It's a little slower, and it's really a kind of a slow, inefficient virus. We've just let this fire burn out of control for a little too long. And it's going to be a big task to actually put it out now.
So how does that compare to, like, you said 12% if someone in your house has it? I feel like if my wife or kid gets the flu, I'm going to get it. Like, there's just no way around it. How about, and chickenpox, when I was a kid, we'd have chickenpox parties and everyone got it that, looked at each other. So how does that compare to something like that?
I think it's actually similar, because I think you remember all the times when you get it. And the other thing with the flu is that 1 out of 4 cases are actually asymptomatic. And so you can get it and not know it, or not know that somebody else has it, and then you've got the flu. And I don't know, it confounds the numbers a little bit. But when they go out and just randomly sample a group of people every week, that's how they figure out these things. And somebody needs to be doing that for, like, if we're going to go back and get kids together in school, I'm okay doing that under a controlled situation where you're testing all the kids before they go back, and then you test them every single week. And so you can actually check, you can see whether they are spreading it, because it may be asymptomatic between most kids, but those kids go home at night. And if you knock out the parents, then there goes your workforce. And there's been a big increase in cases in 20 to 60-year-olds, which is, yeah, right in the middle of the workforce. And so that's what's going to knock out the country, I think, because, yeah, it's very difficult to recover from that. Those are personal lockdowns, and they spread like a virus. Yeah.
I know that our kids not being able to go to school has us on lockdown, knocks out our workforce here.
I know what you mean.
I've got some more questions from parents. Aubrey McGuire, class 08. She was wondering how safe neighborhood pools are.
So the pool itself is fine. The water's not going to get you. The chair's not going to get you, especially if it's outside. The sun is going to irradiate any virus pretty quickly. They are not designed to, not built to withstand any amount of time outside the body very well. They last minutes. Problem is, of course, the other people, because you're splashing around the pool. If there's other people in there, then it brings you into contact with other people, because the pool's kind of a small space, and you can't have a splash fight from, I don't know, 20 feet away. Yeah, that's no good, social distancing. No. So yeah, sure, if you got your own private pool, I guess that's fine, but yeah, not everybody has one of those, I think.
You said that it doesn't last long out, the virus doesn't last long out in the open. Gosh, I remember reading stuff right when this came out that the virus would stay on stainless steel for 2 weeks and plastic for 10 days. I made those numbers up, but for a long time. I'm sorry. So is that that's not the case?
Oh no, that's true. But that's in a lab, and that's yeah, because you're not allowed to work with this virus just out in the sunshine. I mean, you can walk around with the virus, that's legal, but actually working with it, we got a lot of rules to follow, and that's okay. Yeah, I like being safe as much as the next guy. So yeah, they did the best experiment they could at the time, and those are still true. But there's no mechanism that we know of that gets that virus back off the plastic or cardboard or whatever and into your mouth or nose or possibly eyes, we think. Unless you're licking these things, basically, and yeah, just don't do that, because it's weird anyway. Yeah. So
I tell my kids
almost
that.
all person to person rather than surface to person transmission. We think now.
I'm still working on my kids to not lick things.
Yeah. Kids, I don't know. They're the worst. Yeah.
Well, having all that information out there and not knowing what, it's hard to know who to listen to or what to, but I was curious on who are some of the doctors, who are some of the people you follow or reach out to or look for advice during this stuff? Who are people you trust?
Yeah. Most of my information I'm pulling out of scientific papers. So there's a thing called bioRxiv where you get the paper essentially published about 2 weeks before it's really published. And the point is that you can look at it and review it and comment on it. And so, yeah, I tear through those. There's a giant pile of them every day. And believe me, during normal times, if you get one coronavirus paper, in the journal in a month, that's a big deal. But yeah, they are everywhere now. And then let's see, the people that I'm everybody who's got contact with this, like their direct experience is usually pretty interesting, and it'll tell you something. Listen to doctors, you get a particular perspective, but you actually get to kind of go inside the hospital room with them, see what's going on. Listen to research scientists, and you get to find out what their theories are and what they're doing to test it. And you get to also find out about the experiments that didn't work. Like, oh, we thought it might do this. We tried it out. Totally, yeah, it doesn't work that way. And you say, oh, wow, okay, I understand this a little bit better right now. You got the whole coronavirus field. There's, oh gosh, I don't want to name names because I'll leave somebody out, but there's a lot of good people there. There's another good coronavirus scientist at A&M, Julian Leibowitz. I was talking with him last night. And yeah, I think all the coronavirus people are pretty much on the same page on this one. But this is the first time we're kind of holding our meeting outside of the meeting venue. It's being held in the entire world at the same time. And that's a little weird. We're bringing in a lot more people to the group effectively, like everybody. But it's kind of nice as well, yeah.
So trying to look back at some of all the past viruses and looking at these graphs that you can see online, they go up, they peak, they come down, then they go up a couple more times, come back down, and then they kind of it's hard to get anything past that. Is there anything that tells us this is going to go up and stay up and not come down, or is this just,
Yeah.
what timeline are we looking at?
Yeah. There's been a period for, we're about a week past it now, but there was a period of 4 weeks where the US numbers essentially flatlined. They plateaued at a very high number of around 28 to 30,000 cases a day, which is, that's real bad. Yeah. And I didn't ever see any models that predicted that. The models can't be based on this virus because we've never seen it before. So we have to say, well, what if it's like a bacterium? What if it's like polio? What if it's like the flu? Something like that. And even for those, there are so many unknowns there. There are asymptomatic cases for pretty much every disease that's out there. And there are flaws in testing. There are people that don't ever get tested. There are people that die and you never know whether they had it or not. And there are people, every time they do a model, they're looking at some aspect of this and they're trying to figure out what's going on there. So it's not like the whole model is ever gospel, but there's something useful that you can pull out of every one. It's just that the useful part is not where the line ends up and when, unfortunately, which is the only thing anybody really wants to know. Because I want to go back to the world that I knew from 5 months ago. Yeah.
Do you think that just the increased cases or the increased amount of testing? I know that seeing the testing sites just packed with people in line waiting to get tested, weeks ago, and now there's not very, the lines aren't as long. It seems like you could just drive up and get tested.
Yeah. And I think a lot of that is more psychology than virology. When the lockdown, but nuance is not a thing that is easily communicated, I think, in a public setting. So the things that Governor Abbott was saying were, essentially be really cautious, go out if you have to. The stay-at-home order and the go back to some degree of normality order were actually very, very similar. But what got through to people, or seems to have got through to people here in Texarkana, and I was just down in College Station, is that it's over and, do whatever. And the case numbers have really gone up in a lot of places. And I think people think it's over and it's just spreading like wildfire now. We have at least twice as many cases today compared to what we had when the state actually went into lockdown. And we're talking about opening up further, which, from an epidemiological point of view is insane. But I can see why they're trying to do it, because you can't just have a state that does nothing and hemorrhages money indefinitely. But oh my gosh, if we could just do what we're doing in a mask, this would be so much better.
Yeah. And that's what that seems like they've gotten used to that in China, and maybe why they've been able to contain it is that's normal, almost normal way of life. Right. Is when something pops up, they put a mask on.
It's a tradition, exactly. Yeah, it's a thing that is drummed into you that this is what society expects of you. In this country, society expects you to wear an American flag T-shirt and do your own darn thing. Yeah, because this is America. And
you're damn right.
Exactly. Yeah, that doesn't work great in a situation like this.
Well, I know everyone's looking forward to getting back to normal. Hopefully, it's sooner than later.
I don't know if there's going to be a new normal on the other side of this. If we don't get rid of this thing this year, I think what we're doing is it's kind of like quicksand. It's kind of a nonstop set of little pitched battles that we never quite win. And I don't like the sound of that. We're still at the point where we haven't tried all that hard as a nation. You got individual people like in the hospitals that are trying their brains out, but we have not really mobilized. If you look at what maximum human effort looks like, I would say it's probably something like World War II. You got every industry, every person, you got people skipping meat on certain days. You're funneling everything into this one laser focused goal. You're trying all kinds of crazy new things, but really fast. Some of them don't work. Eventually something does. That's what it may end up taking to actually knock this virus down. And I haven't seen anything like that happen yet. But we still have capacity. We still could do more. And so that's where my hope is coming from.
It'll be interesting to see the cost of to the world economy is compared to you know, what it would be, what would the cost be if we didn't do anything?
Oh, it's going to be like nothing we've ever seen before. Yeah, the numbers just keep going up, and people keep saying, "Oh, well, it's, you know, it's no worse than this particular disaster or this particular economic crash." And then we keep passing those milestones, and yeah, we're going to get to the point where it's no worse than, you know, airborne space Ebola or something like that hasn't happened. And at that point, I don't know. I hope we don't go there. I don't want to go there.
Well, I think Germany's soccer leagues are starting today. It seems like they're getting back to they're trying to get back to a normal way of life. What are they doing different? What are some of these other countries doing that are working better than America?
Well, the
other than China and just full lockdown.
Right. You don't have to go full Wuhan, to defeat this thing. Thank goodness. Yeah. No, the German soccer leagues, that's essentially a league in a bottle. They are controlling every bit of movement of these players. They have to stay in the hotel. They may be able to have their significant other with them or their kids, I'm not sure. But they are bringing all the meals in. And I know there was like one coach that wanted to go across the street for something like cigarettes or something. And they found him. And so he had to be quarantined away from the team for 2 weeks. Yeah. That level of strictness, no fans there. And with that and tons of testing, that's something that we could do. And we could start to apply this. Right now, most people have never been tested. And most people probably don't even know where you could go to get tested. And until there is a testing center in your neighborhood that you can go to, and you're going there like every week, but even something like that, that's so much money, so much effort, being funneled into stopping this virus. It's really a shame that this is what it's come to. But there are still ways to dig out of this, and I'm glad there are.
Just forgot what I was going to ask you. Man. Well,
is everything terrible? I don't know, I guess. Yeah.
That's not what we want to hear.
Okay, fine.
We want to hear that June 2nd we can go back to normal. Everything's perfect.
The flowers are blooming just like normal, and I was driving through, and I was very happy to see that over in Central Texas. So. Yeah. Oh,
definitely. I wanted to ask you about antibody tests. Have you taken an antibody test? Are there any good? Are there any good antibody tests that people could take?
Yeah, there are more than 100 that have emergency use approval. That's just the fancy bureaucratic way of saying that they have been tested out. And to get this approval, I think you have to buy a set of 20 tests, and they're all unknowns. And so 10 of them are going to be positive, 10 are going to be negative. They make them in a safe way. And then you got to try your test on those. And if you get something like 19 out of 20 right, then the FDA will say, yeah, okay, you have emergency use authorization, and we'll come back to this and actually look how extremely good your test is later. And so a lot of people are doing this. Most of the tests are real similar. They're all something like a pregnancy test. They just use different mechanisms to find those antibodies in there. The thing with the antibody test, though, is for the most part, it just gives you certainty that, you had it at some point. The antibody test won't give you a positive until about 2 weeks after you get infected, not because the test is bad, but because your immune system is slow and not very good. It just takes a while to build up antibodies that can actually be detected. And so it's nice peace of mind at this point. Now, the antibody test is going to become real important when we actually when we get a vaccine. Because you ought to be able to do an antibody test, not just a general antibody test. That just says you have something that binds to some part of the virus. What you need to know is that you have antibodies that bind to a particular part of the virus that's the key to this virus getting into the cells. And they can do tests that get pretty close to that. There are a couple versions that work like that. And so you could vaccinate people, and then you could keep testing. You get your once a month test. And then when your antibody levels drop below a certain level, you're going to be susceptible again. So maybe in a year's time. And at that point, they know that, okay, antibody levels are down. Yeah, we've got to get you in for your vaccine. And then we'll get you topped right back up again, and you'll be good. So that's where the antibody tests come in. We also need, I would say, improved versions of the test. The test is, like for the actual virus, is just so ridiculously complicated. It involves a thing called a quencher that eats fluorescence from another molecule. It's all this crazy physics involved. And the thing is, you have to heat and cool the whole sample over and over again, like 40 times. And heating and cooling, to some extent, breaks up some of the parts in there. When they break up, then you start to get false positives. And so there's kind of a limit to what you can detect. And sometimes it says that you've got a little bit of virus when you don't actually. Yeah, there's definite room for improvement, but it's pretty good that we have one anyway.
So what do you think the percentage of false positives are on testing?
Pretty low, and it depends where they go. So you rate these things by a thing called cycle time. And so you're doing 40 cycles. And if this thing rises at cycle 10, oh my gosh, yeah, you tested a pile of pure virus. Yeah. If it rises at 20, you've got somebody that has the worst case I've ever seen. 30 is about normal. When you get to 35, that's about the edge of where you would consider it, marginally okay. If you've got people that are coming up at 37, 38, 40, that's right at the end of the test, and some of those are going to be false positives. And so they always have arbitrary cutoffs for these things. They'll say, all right, if you got a cycle number 36 or less, we'll say it's good or good enough. But usually with something like that, it would be better to have a second kind of test that you could do that would actually confirm that, at least for the questionable ones.
All right. Well, what's the number we've got to get we've got to get down to before we can? If it sounds like there's a possibility this will be around for a long time, can keep coming back. It's a 1.5% chance if you get it. What are the goals? What are we trying to get this down to where we can
look at if you look at what China did when they opened up, when they were down below 1,000 cases per day, 1,000 new cases. It was something like 400, 300 new cases a day. And at that point, they were able to open up pretty much completely and do it safely. And their numbers have stayed down. We don't have a lot of people traveling internationally. I think your international airline ticket is going to come with a 2-week stay in a very nice hotel of your choice now in the future. Yeah.
I'm flying to
because
Hawaii.
We'll have to. Yeah, and a rod shoved up your nose, big Q-tip. But yeah, if that's what we have to do to get back to normal without just this thing exploding again, I guess it's uncomfortable and annoying, but 2 weeks in a hotel, that's not too bad. Yeah, maybe we need longer vacation periods at work. Yeah, maybe that's the real end game here.
Well, Dr. Ben, thank you so much for joining us. I know it's a family weekend, Memorial Day weekend. A lot of people are barbecuing. Some people are playing golf, and we really appreciate you taking the time to speak with us.
Well, I got some bacon and eggs cooking in there that are calling my name. I can smell it. Yeah. So,
right.
Yeah.
Well, we got next week. We're talking with Ross Bjork, the athletic director from Texas A&M. We'll find out what the football season looks like and if life can go on in Texas. That's Texas football is more important than just about anything else in the world. So we'll find a lot out next week. So thanks again, Dr. Ben, for joining us. Also, for everybody out there, if there's any other questions you have, you can go to. You've got a Facebook page, Ask Dr. Ben. It's pretty awesome. You take questions from anybody and answer all of them, and it's pretty neat. So thanks for doing that. And
Dr. Ben's Science Group. That's what it's called if you're Googling it. It's still kind of small, but yeah, you're welcome if you want to come in.
All right. Well, thank you very much, Dr. Ben, Bill Hill. Hope you're playing well out there, and we'll talk to everybody next week.
Thanks, Logan. I'm playing, but it is a great day out here. So great job. Enjoyed it, Dr. Ben.
Thank you.
Bye-bye.